Very much. Tēnā koutou katoa. As Phil said, I'm going to talk to you about some work that we've done together looking at bioprotection against fungal pathogens, and particularly we wanted to understand incursions and host range. So won't need to talk about this much because it's been well covered in the last two presentations, but we can think of border biosecurity as a continuum. It starts with what's going on actually outside of our borders, and other studies have shown that there are still plenty of pests and pathogens available to enter New Zealand. And so New Zealand and Australia are the darkest colored lines on this graph, but basically the point is that less than half of the pests and pathogens that could affect plants existing here in New Zealand are already present. So there's lots of pressure coming in from the border. And then, of course, there's what happens actually at the border when things arrive, and then what happens once an incursion occurs and things are inside our borders. How do they spread within the country, and how do they multiply, and what impacts do they have? So we're interested in looking at these last two phases, what happens at the border and what happens inside the border or post-border. And we thought that it's really important to look at these pathogen incursions because it won't have to convince you that pathogen incursions can be really important. We've seen some really interesting work today on Cody dieback, and we've heard a little bit about myrtle rust, and we'll hear more about that. Both of these examples show us that pathogen incursions can have serious impacts on our native biodiversity, on taonga species, and on our economic biodiversity and economic benefits that we derive from plants. So understanding pathogen incursions is really important. The problem is that pathogens are really small, and so they're quite cryptic, and it's hard to track them, it's hard to tell when they arrive, and it takes a lot of effort to actually find out what's going on. But the advantage that we have here in New Zealand is we actually have the opportunity to understand pathogens in a way that's not possible in a lot of other countries. So we're obviously very geographically isolated, and we've also been keeping really good records for the last 150 years or so. So in New Zealand, unlike most countries, we have a pretty good idea of what pathogens are native and what pathogens are alien. And we have much better records on what species are affected by different pathogens and when the pathogens were first found here. And all of that data has been collected and made available by Manaki Whenua. And so our collaborators, Jerry Cooper in particular, has been curating that database. And it is primarily focused on above ground plant pathogens. And there's a heavy emphasis on commercial species because this is where most of the work historically has been done. And this is a database of symptomatic plants. So someone went out, they saw a plant with symptoms, and they determined what that pathogen was, and they've recorded the pathogen and the plant that it was associated with and when that was first observed. And so from that, we can tell when pathogens first arrived in New Zealand or were first detected, and we can also tell what plant species they associate with. And so this is the information that we've used then to look at what's actually happening at our borders. And the first thing we wanted to do is to look at how pathogens arrive over time. We wanted to know whether pathogen arrival is linked to trade or to tourism. So we looked at trade volume and air passenger arrivals. We also wanted to know whether pathogen arrival rates differ between different sectors of the economy, and whether we can see some evidence that our biosecurity efforts are making a difference. And so to do this, we looked at plants that belong in one of four sectors: crop plants, fruit crops, forestry species, and pasture species. And then we also looked at alien pathogens that were first recorded on one of those plants. And then we created a model that accounts for survey effort and the lag between arrival and detection to look at how many pathogen species were arriving on these plants each year across our database. So here's what we found. On the y-axis is the rate of pathogen arrival. So that's how many new pathogen species are showing up in New Zealand, are estimated to arrive in New Zealand in a given year. And then the x-axis on the far graph is import volume, so that's how much is coming into our borders. And then this one closest to me is air passenger arrivals, so this is looking at tourism. And what we can see is that between 1930 and 1980, we have a roughly linear relationship between trade and pathogen arrival rate. More trade, more pathogen arrivals. Each dot represents a particular year, and the red dots show you the decades just to give you a sense of the passage of time. What's interesting is we don't see a similar pattern with air passenger arrivals or tourism. So in that same set of time, from the 1920s to the 1980s, although pathogen arrival rate is changing a lot, air passenger arrival rate isn't changing very much. So we're not getting very many new people coming in, but we're getting a lot more pathogens. Trade actually provides a much better fit during that time period. What's interesting is that in more recent years, that's changed. So since 1990, we're seeing that pathogen arrival rates are relatively constant, but import volume continues to increase, as does air passenger arrivals. And we argue that this is evidence of biosecurity actually working, that as more and more biosecurity measures are implemented, we're able to increase our trade and increase our tourism without substantially increasing the risk that we're exposing ourselves to in terms of incoming pathogens. And so the 1990s, for example, is when stricter quarantine of live plant material was implemented. And it's also when some of the stricter regulations around incoming air passengers were implemented too. And we see that seems to be making a difference, and that we've decoupled trade from pathogen arrivals. Now, we wanted to see how this applied to the different economic sectors that we considered. So this looks at pathogen arrival rate across economic sector. And what you can see is that for crop and fruit trees, pathogen arrivals have been relatively steady over time. For pasture species, there was a big peak in the 1960s and 70s. And then for forestry species, we actually see that pathogen arrival rates are continuing to increase. But as we know from the previous slide, trade imports volume is important. So we wanted to account for that when we looked at pathogen arrival rates. So this shows pathogen arrivals per import volume. And what we can see is that for both crop and pasture species, there's a peak in the 70s, and then arrival rates decrease relative to the amount that's being imported in those sectors. By contrast, for horticultural species, it really hasn't changed much over time, and for forestry species, it continues to increase. Now again, we argue that there's a connection here with biosecurity measures. So in the late 1960s, New Zealand entered the OECD seed certification scheme, which provides for stricter regulation of seed imports. And that's going to be particularly important for crop and pasture species. But it didn't apply to a lot of fruit species. So we see that decline subsequently in crop and pasture, but it didn't have an effect in the fruit tree industry. For forestry, there was inspection of timber imports from about the 1950s, but that was mostly focused on invertebrates. And not only timber imports, but actually pathogens can come in on just wood packaging that's present in a lot of other commodities that are traded. Now, we do have standards around wood packaging, but that's come in quite recently in the 2000s. And our data only goes to 2014. So we think that we haven't seen the impacts of those kinds of biosecurity yet. So it's providing some compelling evidence that biosecurity implemented for crop and pasture species in the 60s, late 60s, early 70s, has decoupled trade and pathogen arrivals. But the lack of similar standards for forestry until recently means that forestry pathogen arrivals continue to increase throughout our data set. So the important things to take away is that incursions are continuing. We're still seeing the arrival of new pathogen species every year here in New Zealand. So this is still something that matters. But even though pathogen arrival is generally linked with trade, biosecurity has broken the link in some sectors. And that really emphasizes the importance of this biosecurity. Now, we also wanted to know what happens once pathogens arrive in New Zealand. What's the next step? Lauren talked about this a little bit this morning, but there's this idea of pathogen spillover. That is, we have both alien and native plants present in New Zealand, and each of them have pathogens, alien and native pathogens. But what can happen is that, for example, if an alien plant is present, hosts a lot of pathogens, those can then move over onto native species that are growing nearby. But we don't understand a lot about how this happens. So we do have some examples. Myrtle rust is a good example of a plant that is present on aliens, can build up into high abundance, for instance, in places where there's a lot of alien myrtaceae that have been planted, but then can spill over onto our native species. There are examples where that happens in reverse as well. So this is an armillaria that's a root rot of pine seedlings, and it's native. So it's native here in New Zealand, but when we plant out these pine seedlings, it can actually cause death, which if you're trying to grow a forestry plantation is a bad thing. If you're trying to control wilding pines, it might be a good thing. But we're interested in how often this happens, how common is it, and can we understand what drives pathogen spillover? And we were looking, again, at a national level using this plant pathogen database. So we had 4 key areas that we thought might influence the rate of these kind of novel associations between alien plants and native pathogens, or native plants and alien pathogens. And so we saw taxonomic similarity as something that's thought to be important. Plant defenses are often taxonomically or phylogenetically conserved, so pathogen will often infect multiple species within a genus. Contact opportunity is how much these plants, how likely these plants are to be planted close together or to occur close together, which should increase the opportunity for the pathogen to jump between them. Pathogen traits may also be important. In particular, we looked at whether pathogens could grow on dead plant material, necrotrophs, or if they relied on living plant material, biotrophs. And we also looked at residence time. How long have these species been present here in New Zealand? What we found is that we looked, the y-axis is the number of novel associations that are being formed, so associations between an alien and a native. And then the x-axis shows whether the focal species is alien or native. Now, the far side are pathogens, and then the side close to me are plants. So what we see is that for pathogens, alien pathogens are slightly more likely to infect a native host than the other way around, although this difference wasn't very strong. But we see a stronger difference with plants. So native plants are far more likely to have an alien pathogen than an alien plant is to have a native pathogen. Now, part of that may just be a numbers game. 70% of the pathogens in this database are alien pathogens. So statistically, it's just more likely that you're going to get an alien pathogen on a native host. But the other thing that's really interesting here is that on average, every native plant had at least one alien pathogen. And actually what we find is about 1/3 of the species in our database form novel associations, and that about 10% of all the associations in the database are between an alien and a native. So this isn't uncommon. This is actually happening a lot. And it's happening particularly to our native plants. The other thing we looked at, as I said, is we looked at whether taxonomic similarity was important. So this is for an alien pathogen. On the y-axis is the proportion of all its recorded hosts that are native. So the x-axis then looks at what are the alien hosts of an alien pathogen, and then tallies up how many species are present in New Zealand within those genera, and how many of those species are native and how many are alien. So if you infect a genus of plants that has a lot of native species, you're more likely to have native hosts. And that makes sense, but basically what it's showing us is that these alien pathogens are more likely to infect close relatives of their alien hosts. So that taxonomic link is important. We didn't find that the other traits we looked at were particularly important. So we found this taxonomic similarity really matters. If you share, if a native plant is in the same genus as an alien plant, it's more likely to share pathogens. But our measures of contact opportunity and pathogen traits and even residence time weren't that important. What was important is sampling effort. And this is important to mention. It's sort of obvious, the more you look, the more you find. But we do see in our database that sampling effort has decreased in recent decades, and often that's tied into availability of funding and resource priority. The problem is we don't know what we don't study, and if we're not looking, then we're not finding out what's going on. And it just really emphasizes the importance of actually continuing that monitoring post-border so that we know what plants are being affected, which pathogens are spreading, and what kind of impacts those might have. Now, the last thing we did is we actually put all of these into a network, and I'll just show you this very briefly. So the green circles are native plants. The squares represent pathogens. On the far side, those squares are alien pathogens, and on the near side, these squares represent native pathogens. The gray lines between a plant and a pathogen show an interaction, an association between them. The thicker the line, the more often that association has been recorded. The bigger the square or the circle, the more often that species shows up in our database. And basically what we see here is that we have a lot of native plant and native pathogen interactions, but there's a pretty significant number of alien pathogens that are affecting our native plants as well. And this is basically another way of looking at these questions. What we find is similar to what we saw when we looked at a species by species basis, that alien pathogens are more generalist. They connect across the network in ways that connect plants, and it's different from the way native pathogens do. And our concern here is that the way those connections are happening may actually increase the likelihood of outbreaks and destabilize this network of interactions. So, what we found is that pathogens continue to arrive, but biosecurity is making a difference in breaking the link between trade and pathogen arrivals. About 30% of all hosts and pathogens have some form of novel association, so this is pretty common. The likelihood that an association between an alien pathogen and a native plant will form does depend on how closely related that native plant is to alien plants already here. But alien pathogens are more generalized than native pathogens, and that changes the network structure in ways that may destabilize the network and make it more prone to outbreaks. So, why does this matter? It shows us that biosecurity and surveillance are worthwhile. Biosecurity is making a difference, and surveillance helps us to know what kind of impacts these pathogens are having and where those impacts are occurring. Alien plants that have a lot of native congeners, a lot of native species in the same genus, are going to be at the greatest risk of creating these pathogen spillover dynamics. So particularly where they invade our native ecosystems, this pathogen spillover is something we should be paying attention to. We also see that as networks change with the arrival of alien pathogens, these dynamics may become less stable and more prone to outbreaks over time. And with that, thank you very much.